The human brain runs on roughly 20 watts of power. That is less energy than a dim incandescent light bulb. The most powerful supercomputer ever built consumes tens of megawatts just to stay operational, with most of that budget going not to computation but to cooling. That is an energy gap of roughly 1 million to 1. If the brain were simply doing the same class of computation as a silicon chip, only in biological material, that ratio would be physically impossible. You cannot run the same calculations on 1/1,000,000th of the energy. The math does not allow it. This means one of two things is true. Either we fundamentally misunderstand what computation is, or the brain is doing something that our computers are not designed to do, something that operates by different physical rules entirely. The evidence, accumulating across multiple independent fields over the past 2 decades, increasingly points toward option 2. And understanding what that something actually is has become one of the most consequential scientific projects of our time. We have spent decades trying to simulate the human brain using conventional computers. The strategy has always been the same. Add more transistors, increase clock speed, expand memory, and assume the gap will eventually close. It has not closed. In fact, the more precisely we attempt to model the brain, the more clearly the mismatch becomes. The problem is not that our computers are not fast enough. The problem is that they are operating according to the wrong physical rules. To understand the scale of what we are attempting to replicate, most people dramatically underestimate the number that matters. The brain contains around 86 billion neurons. That is an enormous figure. But the neurons themselves are not where the complexity lives. The complexity lives in the connections between them. Each neuron links to thousands of others through structures called synapses. When you map the full network of those connections, what researchers call the connectome, you arrive at approximately 100 trillion distinct synaptic contact points. 100 trillion. There are roughly 10 times more synaptic connections in a single human brain than there are stars in the Milky Way galaxy. And none of those connections are static. Every second, hundreds of trillions of synapses are either strengthening or weakening based on incoming chemical signals. The brain is not a fixed circuit. It is a living, continuously reconfiguring network that never stops moving. Attempting to simulate that level of dynamic, massively parallel activity on a machine that processes information sequentially is something like trying to capture the ocean with a series of still photographs. You can approximate it, but you will always miss the motion. The most serious classical attempt to directly address this problem was the Blue Brain Project, launched in 2005 at the École Polytechnique Fédérale de Lausanne. The goal was to build a biologically detailed digital reconstruction of the mammalian brain from the ground up, starting small and scaling up. Using some of the most powerful supercomputers available at the time, the team succeeded after years of effort in simulating a small neocortical column from a rat brain, a sliver of tissue containing roughly 30,000 neurons. To run that simulation, they had to track every ion channel, every electrical spike, and every chemical gradient across all 30,000 cells simultaneously. The computational cost was staggering. That was 30,000 neurons. A human brain has 86 billion. When researchers tried to project what it would take to scale the simulation to a full human brain at the same level of biological detail, the numbers became unreachable. You would need computational resources on the scale of a large industrial facility just to store the data, let alone process it in anything approaching real time. Classical hardware had hit a wall that no amount of chip fabrication would dissolve. The depth of that wall became vivid in 2013, when a team used Japan's K supercomputer, one of the most powerful machines on the planet at the time, to simulate just 1 second of brain activity. The simulation represented only about 1% of the full human brain. After the researchers loaded in the model and started the calculation, the K computer required 40 minutes of processing time to generate that single second of neural activity. 40 minutes of supercomputer time to recreate 1 second of biological thought. That is not a simulation. That is an extraordinarily slow motion replay of a system running thousands of times faster than the machine trying to capture it. More recent work has made genuine progress. In 2025, a team used the Fugaku supercomputer, which succeeded the K computer as Japan's flagship machine, to simulate the mouse cortex with nearly 10 million neurons and 26 billion synapses, achieving performance approaching real time. This was a meaningful milestone, but it was made possible by simplifying the biological model and running it on hardware that cost billions of dollars to build. And even at that scale, the mouse cortex represents a small fraction of the complexity of a human brain. The gains in classical computing have been real. The ceiling is still not high enough. What makes this problem fundamentally difficult is not just the number of neurons. It is the architecture. A classical computer is a linear machine. Data moves from storage to processor, gets calculated, and moves back. Even with parallelism, the underlying structure is flat. The brain is not flat. It is a three-dimensional, fully interconnected mesh in which every part of the network can influence every other part simultaneously. Running a three-dimensional, interconnected biological network on a two-dimensional, linear architecture is not an engineering problem that can be solved by building more processors. It is a structural mismatch at the level of physics. The energy problem reveals something deeper. When you look at a modern supercomputer facility, most of what you are looking at is not computing. It is cooling. These machines generate so much waste heat that they require industrial cooling towers and a continuous supply of water just to keep the processors from destroying themselves. The actual computation accounts for a small fraction of the total power budget. Most of the energy goes into fighting heat. The brain generates almost no waste heat. If you held your hand near a thinking human brain, you would not feel warmth radiating from it the way you would from a running processor. The brain solves complex problems, generates language, processes vision, coordinates movement, manages emotion, and sustains what we call consciousness, all on a metabolic budget that your body maintains through an ordinary diet. 20 watts continuously for decades. In classical computing, heat is a fundamental consequence of making decisions. When a processor switches a transistor from 0 to 1, it forces electrons through a resistive gate. That physical act generates friction, and friction generates heat. The more decisions the machine makes, the more heat it produces. This relationship between computation and thermodynamics is formalized in something called Landauer's principle, proposed by physicist Rolf Landauer in 1961. The principle states that erasing a single bit of information requires a minimum amount of energy to be released as heat. Every time a computer discards a possibility to reach a conclusion, it pays a thermodynamic price. You cannot avoid this in classical computing. It is baked into the physics. The brain does not appear to pay this price in the same way. One reason may be that quantum computation can, in theory, be reversible. A reversible computation leaves no information behind, erases nothing, and therefore generates no mandatory heat release. The brain does not achieve perfect reversibility, but its efficiency suggests it is operating closer to that physical regime than any silicon chip ever has. This points toward a fundamentally different physical mechanism underlying the biology. And the most compelling candidate, the one that has been quietly accumulating experimental support for years, is quantum mechanics. For a long time, suggesting that the brain might operate on quantum principles was treated as fringe science. The objection seemed decisive and simple. Quantum states are extraordinarily fragile. To maintain quantum coherence, a system typically needs to be isolated from its environment, cooled to temperatures near absolute zero, and shielded from all external vibration and electromagnetic interference. The brain is warm, wet, chemically chaotic, and constantly bombarded with thermal noise. The assumption was that any quantum state inside a neuron would collapse within femtoseconds, far too fast to play any meaningful role in cognition. Then scientists started looking more carefully at what is actually inside the neuron. For decades, Neurons were understood primarily as signal cables. The cell membrane carried the electrical impulse, and the interior was thought to be essentially just molecular plumbing. That picture has changed. The interior of a neuron is packed with a dense structural scaffold called microtubules. These are hollow, cylindrical protein structures that form the skeleton of the cell and are found throughout biology. In most cells, they are pure scaffolding. Inside neurons, they may be doing something more. Physicist Roger Penrose and anesthesiologist Stuart Hameroff proposed a theory they called orchestrated objective reduction, or Orch-OR. They argued that microtubules are not merely structural supports. The geometry of the protein chains forming the microtubule wall and the arrangement of electrons within those chains may create a partially insulated quantum environment inside the tube. If correct, this insulation would allow quantum states to persist long enough inside the neuron to participate in information processing. The microtubule would function as a biological quantum waveguide, shielding coherent quantum states from the thermal noise of the surrounding cellular environment. The scientific community was skeptical, and the skepticism was reasonable. But the evidence has been accumulating. Researchers have observed quantum vibrations in the protein lattice of microtubules. They have found that energy propagates through these structures with anomalously low loss, behaving more like a coherent wave than a classical thermal disturbance. Studies have linked this propagation to networks of tryptophan molecules arranged inside the tube. When these molecules resonate together, they appear to generate a macroscopic quantum state, similar in principle to how a laser works. In a laser, individual photons synchronize into a single coherent beam through stimulated emission. Inside microtubules, molecular resonances may synchronize into a single coherent quantum state spanning the length of the structure. This is not proof that the brain runs on quantum mechanics, but it is no longer dismissible as speculation, and it changes the conversation about simulation entirely. If quantum processing is happening inside neurons, even at a limited scale, then a classical computer cannot faithfully replicate it. A binary switch cannot simulate a quantum superposition. These are not different speeds of the same process. They are different physical categories. A simulation that ignores the quantum layer of the brain is not a simulation of the brain. It is an approximation of certain statistical behaviors the brain produces, which is a fundamentally different thing. To understand what quantum computers actually offer, it helps to be precise about what makes them different. A classical bit is always either 0 or 1, with no ambiguity. A quantum bit, called a qubit, can exist in a superposition of 0 and 1 simultaneously. This is not a metaphor or a convenient shorthand. The qubit is genuinely in both states at once until it is measured. When you build a system of multiple qubits, they can also become entangled, meaning the state of one qubit is instantaneously correlated with the state of another, regardless of physical distance. They are no longer independent. They are a single unified quantum system. Measuring one instantly determines the state of the other. This is where the power of quantum computation originates. When you solve a problem with classical bits, you explore possible solutions one at a time or in parallel batches, but always moving through discrete, definite states. When you solve a problem with qubits in superposition, the machine is exploring all possible solutions simultaneously through quantum interference. The probability amplitudes of wrong answers cancel each other out, while the amplitudes of correct answers reinforce each other. The computation does not guess and check. It converges on the answer through the physics of wave interference. For simulating physical systems like the brain, this matters in a specific and decisive way. To model any physical system, you need to describe its energy state. In physics, this description is called the Hamiltonian. The Hamiltonian is essentially the energy fingerprint of a system. It captures how every component vibrates, interacts, and evolves over time. A single neuron has a Hamiltonian. A network of neurons has an incomprehensibly more complex one. If you can define the Hamiltonian accurately and simulate how it evolves, you have simulated the system. When a classical computer tries to simulate a Hamiltonian, it solves the mathematics explicitly, computing the differential equations that describe the evolution of the energy state one step at a time. As the system grows, the equations grow exponentially harder. At the scale of even a modest neural circuit, the classical computer essentially freezes. It is trying to describe every ripple in a pond by tracking every water molecule individually. A quantum computer does not try to solve the equation. It tries to become the equation. This is the concept of quantum simulation. You configure the qubits so that they are governed by the same physical laws that govern the neurons. You set up the interactions between qubits to mirror the interactions between biological molecules. You are not calculating what the neuron will do. You are building a quantum replica of the neuron and observing what it does. The hardware and the simulation are the same thing. This idea goes back to Richard Feynman, who articulated it precisely in the early 1980s. He observed that nature is not classical. Nature is quantum. Therefore, if you want to simulate nature, you need a quantum machine. For 40 years, that prescription sat waiting for hardware capable of fulfilling it. The practical bottleneck until recently was error. Qubits are extraordinarily sensitive to their environment. A stray magnetic field, a temperature fluctuation of a fraction of a degree, a cosmic ray passing through the chip, any of these can cause a qubit to flip from its correct state to an incorrect one. These are quantum errors, and for years they accumulated faster than any algorithm could run. The computers were powerful in theory and unusable in practice. The field of quantum error correction existed to address this, but it faced a cruel paradox. Adding more physical qubits to implement error correction codes also added more potential failure points. The system became noisier as it grew larger. Errors were being fixed at the bottom, while new ones were introduced at the top. This changed fundamentally in late 2024 with the announcement of Google's Willow quantum chip. Willow was not simply a faster processor. It was a demonstration that the error correction paradox could be broken. The Google team showed that by grouping physical qubits into logical qubits, clusters of physical qubits that collectively protect a single unit of quantum information, the error rate decreased exponentially as they added more physical qubits to each logical group. For the first time, making the system larger made it more stable rather than less. Adding more physical qubits exponentially reduced errors rather than amplifying them. This is the turning point. It is the proof of concept that quantum computers can, in principle, maintain stable quantum states for the duration of a complex computation. For biological simulation, this matters enormously. The brain maintains a continuous state of activity for decades. Simulating it requires a computer that can sustain coherent computation not for microseconds, but for minutes and eventually hours. The Willow result suggests that is achievable. We are moving into an era where quantum algorithms can run long enough to observe how a digital neural network evolves and adapts over time. The immediate practical applications are in medicine, specifically in understanding and treating neurodegenerative disease. Diseases like Alzheimer's, Parkinson's, and Huntington's are not simply diseases of neural death. They are, at their molecular foundation, diseases of geometry. The nervous system relies on proteins, and proteins must fold into precise three-dimensional structures to function. A protein begins as a long chain of amino acids. To become active, this chain must collapse into a specific shape determined by the interactions between its chemical groups. If it folds correctly, it does its job. If it folds incorrectly, the misfolded protein can become toxic, clumping into aggregates that damage and kill neurons over decades. The challenge of predicting how a protein will fold is one of the most computationally demanding problems in science. A modest-sized protein can theoretically adopt more distinct conformational shapes than there are atoms in the observable universe. A classical computer approaches this by guessing a shape, calculating the associated energy, adjusting, and guessing again. This brute force search can take months on a supercomputer for a single complex protein. The field changed significantly when DeepMind released AlphaFold, an artificial intelligence system trained on known protein structures that could predict folding with remarkable accuracy. But AlphaFold is a pattern-matching engine. It works by learning from examples. For novel proteins with no structural analogs in the training data, it struggles. And for simulating how misfolding happens dynamically in real time under specific molecular conditions, it is not the appropriate tool. A quantum simulation approaches protein folding from physics rather than pattern matching. In nature, a protein folds by seeking the configuration that minimizes its energy. The chain samples different shapes continuously and gravitates toward the state of lowest energy, the stable configuration. A quantum computer can simulate this process directly. The qubits settle into their own state of lowest energy automatically through the physics of quantum mechanics, physically mirroring the folding process the protein undergoes. The computer does not search through possibilities. It physically enacts the folding. Combine this with the concept of a digital twin, and the implications for medicine become concrete. Imagine scanning the molecular structure of a specific patient's brain and loading that data into a quantum processor tuned to reflect that patient's neurochemistry. You have built a sandbox replica of that person's neurobiology. You can introduce a candidate drug molecule into the simulation and observe how it interacts with the misfolded proteins in real time. You can watch whether it interrupts the aggregation cascade. You can detect unexpected interactions with other molecular systems before the drug has ever entered a living body. You can run thousands of variants of the compound in a single afternoon. This compresses a development timeline that currently spans over a decade and costs billions of dollars into something measurable in weeks. Beyond medicine, there is an application emerging in brain-computer interfaces, and it involves a problem that has defined and limited the technology since it began: latency. When a person using a neuroprosthetic decides to move a hand, the brain generates a cascade of electrical signals encoding the intention. A classical computer receiving those signals must filter the noise, run a decoding algorithm to interpret the pattern, generate a command, and send it to the motor device. Each step takes time. The result is a noticeable delay between intention and action. The user does not feel like the prosthetic is part of their body. They feel like they are operating a tool with their thoughts. Neural signals are not clean binary messages. They are probabilistic clouds of electrical activity distributed across many neurons simultaneously, riding on a background of biological noise. A classical decoder must wait until the signal achieves sufficient statistical clarity before it can commit to an interpretation. It discards ambiguity because ambiguity breaks the logic of binary decision-making. This waiting is where the delay lives. A quantum processor does not need to wait for ambiguity to resolve. A qubit can natively represent a probability distribution rather than forcing a signal into a binary category. The raw, noisy probability field of the neural signal can be mapped directly onto the qubits, which process the entire distribution at once. The quantum system can decode the user's intention before the biological signal has even fully propagated through the neural pathway. This speed is the difference between a prosthetic that requires conscious effort to operate and one that responds as naturally as a biological limb. It is the difference between using a tool and having a body part. The implications extend further than motor control. Motor signals are relatively simple because they encode specific muscle commands with a limited vocabulary. Abstract thoughts are not. When you visualize a face, that visual pattern is distributed across dozens of cortical regions simultaneously. The information is encoded in the correlations between regions, not in any single stream of data. Classical algorithms struggle with this because the number of variables is too high and the correlations are too subtle. A quantum system can analyze correlations across many brain regions in parallel, detecting the subtle synchronizations that represent specific memories, images, or concepts. This opens the possibility of decoding semantic thought directly, not just motor intention. If the latency is low enough and the decoding accurate enough, the interface stops being an interface. The machine becomes a transparent extension of cognition. Communication does not require speech or gesture. The boundary between the biological mind and the digital processor becomes functionally invisible because information flows between them at biological speed. Quantum computers today are still relatively small. The largest systems have hundreds of logical qubits, sufficient for demonstrating principles and solving narrow problems, but far short of what is needed to simulate even a simple neural circuit in full biological detail. The engineering challenges of scaling up are formidable. Maintaining quantum coherence across thousands or millions of qubits, while keeping error rates below the threshold required for accurate simulation, will require years of incremental progress. The trajectory is toward a hybrid architecture. Future computational systems will almost certainly combine classical and quantum processors in a division of labor that exploits the strengths of each. The classical processor handles what it does best, the stable, structured, logical framework of the computation. It manages input and output, maintains the static map of the connectome, handles the bookkeeping. The quantum coprocessor handles what only it can do, the nonlinear, probabilistic, entangled simulation of dynamic neural activity. One system holds the skeleton, the other breathes life into it. This hybrid approach is likely how the first real-time simulation of a cortical column will be achieved. A cortical column is the basic repeating unit of the cerebral cortex, a cylindrical bundle of neurons about 2 millimeters tall and roughly half a millimeter in diameter, containing tens of thousands of cells organized into 6 distinct layers. It is the fundamental building block of higher cognition. Simulating one column in real time with full biological fidelity would be a transformative milestone. It would give researchers a living model of the core computational unit of the human brain. From there, scaling to larger regions becomes a matter of engineering rather than fundamental discovery. There is a deeper question lurking underneath all of this, one that cannot be deferred indefinitely as the technology matures. It is the question of consciousness itself. If we build a quantum simulation of a human brain that accurately replicates the energy states, the protein dynamics, the firing patterns, and the quantum fluctuations of biological neural activity, what exactly have we built? Have we built a model that calculates data, or have we built something that experiences the calculation? This is what philosopher David Chalmers calls the hard problem of consciousness. The easy problems of consciousness, in his terminology, are the ones that can in principle be solved by good enough neuroscience. We can explain how the eye encodes visual information, how the hippocampus consolidates memory, how the prefrontal cortex evaluates decisions. These are difficult but tractable scientific problems. The hard problem is different. It is the question of why any of that feels like something. Why does the specific pattern of activity associated with seeing red produce the subjective experience of redness? Why is there something it is like to be a brain? No equation in current physics answers this. There is no term in any Hamiltonian that corresponds to the feeling of sadness or the taste of sugar. The subjective dimension of experience exists outside the current scope of physical theory. Quantum mechanics offers an intriguing new angle on this problem. Integrated information theory, proposed by neuroscientist Giulio Tononi, argues that consciousness arises from the degree to which a system is irreducible. A conscious system is one where the whole is more than the sum of its parts, where the interactions between components generate information that cannot be decomposed into the sum of each component's individual contribution. The measure of this irreducibility is called phi. In a classical computer, components are separable. You can isolate one processor from another. Information flows between them, but each component is describable independently. In a quantum computer, entangled qubits are not separable. They form a unified wave function. You cannot describe the state of one qubit without describing the state of the whole entangled system. This physical unity corresponds to a high degree of irreducibility. If integrated information theory is correct, an entangled quantum system would naturally tend to exhibit a meaningful value of phi. Whether this is sufficient for consciousness remains unknown, but it represents a more credible substrate for integrated information than anything classical computing provides. The question carries immediate ethical weight. If we create a quantum simulation of a human mind for the purpose of testing a drug, and that simulation generates signals consistent with pain, are we ethically obligated to treat those signals as meaningful? In a classical simulation, dismissing them as mere code is defensible. The classical computer is printing the words. The word pain and the state of pain are clearly different things. In a quantum simulation that is physically isomorphic to a brain in distress, the distinction becomes far less clear. The physical state of the machine may be, in some morally relevant sense, the same as the physical state of a person suffering. This is not a distant philosophical puzzle. It is an engineering deadline. As quantum simulations become more detailed and more biologically accurate, the scientific community will need a framework for evaluating the moral status of simulated systems before the technology makes the question urgent. We do not currently have such a framework. We do not even have consensus on what features of a physical system give rise to experience in the first place. To understand why quantum mechanics is the right physical language for the brain, it helps to understand the scale at which biology actually operates. We tend to think of biology as chemistry, and chemistry as something that happens at a level comfortably above quantum physics. That picture is wrong. The molecular machinery inside every cell operates at length scales where quantum effects are not peripheral. They are central. A water molecule is about 3 angstroms across. An angstrom is 1/10,000,000th of a meter. The synaptic cleft, the gap across which neurotransmitters travel to transmit signals between neurons, is about 20 nanometers wide. At these dimensions, the behavior of electrons is governed by quantum mechanical wave equations, not by the classical laws that describe billiard balls or planetary motion. The proteins that open and close ion channels in a neuron's membrane are molecular machines operating in this quantum regime. When a voltage-gated sodium channel opens to allow an action potential to propagate, the conformational change in the protein that opens the channel is driven by the quantum mechanical behavior of electrons and atomic nuclei. Classical mechanics cannot accurately describe it. It was never designed to. This is important because it means the brain is not a system that happens to be implemented in biology. It is a system whose fundamental operations are quantum mechanical. The neurons and synapses we observe at the scale of cellular biology are the emergent high-level output of processes that, at their foundation, are as quantum as any phenomenon studied in a physics laboratory. To simulate a quantum system on a classical computer, you need to track the amplitude of every possible quantum state of the system. For a system of a certain number of quantum particles with 2 possible states each, the number of amplitudes you must track grows exponentially with the number of particles. For even a modest quantum system, this number rapidly exceeds any conceivable classical memory. A system of 300 entangled qubits would require more amplitudes to describe classically than there are atoms in the observable universe. You cannot fit the description of such a quantum system into any storage medium made of matter. The brain contains vastly more than 300 quantum-relevant degrees of freedom. If even a small fraction of neural processing involves quantum entanglement and coherence, classical simulation is not just difficult. It is formally impossible at any scale of hardware that could ever be built. The only machine that can simulate a quantum system of that complexity is another quantum system of comparable complexity. This is Feynman's prescription made concrete. What distinguishes quantum computation from classical computation is not the hardware. It is the class of logical operations that the hardware can natively perform. A quantum processor can natively perform operations that are impossible on any classical machine, regardless of how fast the classical machine runs. For brain simulation, the relevant native operations are quantum simulations of physical Hamiltonians. A classical processor cannot perform these operations natively. It can only approximate them through numerical methods that become exponentially expensive as the system size grows. A quantum processor performs them directly. This is not a matter of degree. It is a matter of category. The implication is that a quantum simulation of the brain is not just a better version of what classical simulators have attempted. It is a different kind of thing entirely. Classical brain simulations are models in the conventional scientific sense. They are mathematical descriptions of how we believe the system behaves, encoded in computational form. A quantum simulation of the brain aspires to be a physical instantiation of the brain's dynamics. Not a description of what the brain does, but an enactment of it. The language of the simulation and the language of the biology are the same language. The first complete connectome ever mapped at the level of individual neurons and synapses was the connectome of Caenorhabditis elegans, a tiny roundworm with 302 neurons and approximately 7,000 synapses. This project was completed in the 1980s and took over a decade of manual reconstruction from electron microscopy images. With that complete map, researchers could simulate the worm's nervous system classically, and the simulations were informative. But they also revealed something important. Even with the complete connectivity map, the classical simulation could not fully reproduce the worm's behavior. Something was missing. The map was necessary but not sufficient. The missing elements are the dynamic properties of the neurons themselves, their intrinsic excitability, the kinetics of their ion channels, the spatial distribution of inputs across the dendritic tree, and the probabilistic nature of synaptic transmission. All of these properties are quantum mechanical at their foundation. A classical simulation can parameterize them, meaning it can represent them with equations fitted to experimental data. But parameterization is not simulation. It replaces the physics with a model of the physics, and the model is always an approximation. The approximation is acceptable for some purposes. For others, particularly those involving the subtle quantum effects that may underlie phenomena like memory consolidation and consciousness, the approximation may discard exactly the information that matters. The Human Connectome Project, launched in 2009 with funding from the United States National Institutes of Health, set out to map the large-scale connectivity structure of the human brain using diffusion MRI tractography. This technique traces the pathways of white matter fiber bundles connecting different brain regions. The resulting macro-scale connectome has been used to build computational models that capture some aspects of large-scale brain dynamics, but it cannot simulate the neural activity itself. It models statistical patterns at a resolution far coarser than the actual neural dynamics. The macro-scale connectome provides the architectural blueprint. The quantum simulation provides the means to execute the blueprint faithfully. These are not competing research programs. They are addressing different levels of description that will ultimately need to be integrated. There is also a dimension of this problem that is rarely discussed: the role of glial cells. For most of the history of neuroscience, glial cells were considered to be support staff, providing structural scaffolding, supplying nutrients, and maintaining the ionic environment that allows neurons to fire. Research over the past 2 decades has overturned this picture. Glial cells, particularly the astrocytes, are now understood to actively participate in neural signaling. Astrocytes form intimate contacts with synapses. A single human astrocyte can enwrap as many as 2 million synapses. At each synapse, the astrocyte monitors the chemical environment and releases its own signaling molecules, called gliotransmitters, which modulate synaptic strength. Astrocytes also propagate calcium waves through connected networks at speeds and over distances that are relevant to neural computation. The brain has as many astrocytes as it has neurons. A simulation that ignores astrocytes is ignoring half the computational substrate. The calcium dynamics inside an astrocyte involve the same quantum mechanical protein behaviors that govern ion channel opening in neurons. The release of gliotransmitters involves the same quantum-level conformational changes in molecular machines. A complete simulation must model not just the neurons, but the entire cellular ecosystem of the brain. One area where the near-term scientific payoff is most concrete is the simulation of anesthetic mechanisms. Anesthetics are among the most widely used drugs in medicine, yet the mechanism by which they render a patient unconscious is still not fully understood at the molecular level. We have been routinely inducing unconsciousness in surgical patients since the 1840s, and we still do not know exactly how it works. What we do know is that anesthetics interact with proteins in neural membranes. Roger Penrose and Stuart Hameroff have argued specifically that anesthetics act on microtubules, supporting their theory that microtubules are involved in consciousness. If anesthetics disrupt quantum processing in microtubules, this would explain why they eliminate consciousness without simply killing the neurons. The neurons survive anesthesia. Their electrical activity continues to some degree. What disappears is the integrative experience. A quantum simulation of microtubule dynamics under anesthetic exposure would directly illuminate the physical mechanism of consciousness. It would also explain why patients wake up when the anesthetic clears. The quantum processing in the microtubules resumes. This hypothesis is testable, and testability is what makes quantum simulation of biological systems scientifically valuable. To simulate these dynamics requires not just quantum hardware, but also advances in how quantum computers are programmed and controlled. Quantum algorithms for simulating molecular dynamics, including variational quantum eigensolvers and quantum phase estimation algorithms, have been developed and tested on small systems. They work. The results match what we expect from classical quantum chemistry calculations. The limitation is that the systems they can currently handle are small, on the order of dozens of electrons in a molecular orbital structure. Scaling these algorithms to handle proteins with thousands of atoms requires hardware that does not yet exist at the required scale, but the algorithms themselves are ready. There's also a growing body of work on quantum machine learning algorithms, specifically relevant to decoding neural activity. Classical machine learning has been successfully applied to decode motor intentions from neural recordings, but classical decoders face fundamental limitations in the quality and speed of decoding complex cognitive states. Quantum machine learning algorithms can in principle find patterns in high-dimensional data that classical algorithms cannot detect efficiently. Applying these to the high-dimensional probability distributions of neural activity could dramatically improve the quality and latency of brain-computer interface decoding, even before a full brain simulation is achieved. The interaction between quantum computing and neuroscience is therefore not a single monolithic project. It is a collection of converging research programs, each addressing a specific aspect of the grand challenge, each capable of delivering meaningful results at different points along the timeline. Error correction research advances the stability of quantum hardware. Quantum chemistry algorithms advance our ability to simulate molecular-level neural processes. Quantum machine learning advances brain-computer interfaces. Foundational research into quantum effects in biological systems advances our understanding of whether and how the brain itself uses quantum mechanics. All of these programs are active today. Modern artificial intelligence systems, particularly the large language models that have become prominent in public discourse, are trained by exposure to data. They learn statistical patterns in text, images, or other inputs and generate outputs that reflect those patterns. They are extraordinarily sophisticated pattern matchers. They can produce fluent text, recognize images, write code, and generate plans. They are also brittle in ways that biological intelligence is not. They fail at problems requiring genuine reasoning outside their training distribution. They lack the flexible generalization that characterizes human cognition. The reason for this brittleness is architectural. These systems are trained to minimize a loss function over a training dataset. Everything they know is derived from what they have seen. They have no way to engage with novel problems by reasoning from first principles, because first principles require access to the underlying physics of the world, which is quantum mechanical, and which cannot be captured in a data set of text and images. A quantum simulation of neural dynamics is a different kind of thing. It is not trained on data. It is built from physics. Its behavior emerges from the same equations that govern the actual biological system it models. If the model is sufficiently accurate, it does not just replicate what we know. It can explore what we do not know because it is running the same physics that the brain runs. It can discover things that are not in any data set because it is operating from the same first principles as nature. This distinction between statistical pattern matching and physical enactment is fundamental. An artificial intelligence system can tell you things consistent with patterns in its training data. A quantum neurosimulation can do experiments. The history of science is full of examples where a powerful new instrument opened a window onto phenomena that were previously invisible. The telescope revealed the moons of Jupiter, overturning the Aristotelian picture of perfect celestial spheres. The microscope revealed the cellular structure of living tissue, overturning the humoral theory of disease. The functional MRI scanner revealed the large-scale patterns of neural activity during cognition, overturning the localizationist view that individual cognitive functions were confined to small, fixed brain regions. A quantum brain simulator would be an instrument of this same category. It would reveal aspects of neural dynamics that are currently invisible to all existing tools. The progression of milestones that researchers are working toward follows the logic of the underlying physics and the constraints of what hardware can currently do. The first milestone, already being demonstrated in laboratory settings, is quantum advantage for specific quantum chemistry problems relevant to biology. Quantum advantage means showing that a quantum computer can solve a problem beyond the practical reach of any classical computer, regardless of how long the classical machine runs. For quantum chemistry, the target problems involve calculating the ground state energy of molecular systems, the same calculation that underlies protein folding and drug receptor binding. The variational quantum eigensolver algorithm has been used to calculate the ground state energies of small molecules on existing quantum hardware. The goal in the near term is to scale these calculations to molecules with dozens or hundreds of electrons, which is the regime relevant to the active sites of biological proteins. The second milestone is quantum simulation of a biologically realistic ion channel. Ion channels are the molecular switches of the nervous system. They are transmembrane proteins that open and close in response to voltage or chemical signals, allowing specific ions to flow across the cell membrane. This flow generates the electrical signals that neurons use to communicate. An ion channel is small enough, on the order of a few hundred amino acids, to be within reach of near-term quantum simulation. An accurate simulation of an ion channel would be directly scientifically valuable because many neurological diseases are caused by mutations in ion channel genes, and many drugs work by blocking or modulating ion channel activity. The third milestone is the quantum simulation of a synaptic release event. When a nerve impulse reaches the end of an axon, it triggers the release of neurotransmitter molecules from small vesicles into the synaptic cleft. This process involves a cascade of molecular events, including the activation of voltage-gated calcium channels, the binding of calcium ions to proteins called snares, the conformational change of the snare complex that pulls the vesicle membrane into contact with the cell membrane, and the fusion of the two membranes that releases the neurotransmitters. Each step involves quantum mechanical processes in proteins. A quantum simulation of a single synaptic release event would, for the first time, give us a physically accurate picture of how the most fundamental unit of neural communication actually works. Beyond these specific milestones, there are broader architectural developments that will enable larger-scale neural simulations. One of the most important is the development of quantum networks, systems in which multiple quantum processors are connected through quantum communication channels. The brain's computational power comes not from any single neuron, but from the network. A quantum simulation of the brain will similarly need to be distributed across a network of quantum processors, each simulating a local region of the brain, connected by quantum channels that allow entanglement to be maintained across the whole system. Quantum networking is itself an active area of research, driven in part by applications in quantum cryptography and distributed quantum computing. Coherence times have been increasing across different qubit architectures, including superconducting circuits, trapped ions, and photonic systems. Each architecture has different strengths and weaknesses. Superconducting qubits are fast but require cooling to near absolute zero. Trapped ion qubits have longer coherence times but operate more slowly. Photonic qubits are naturally suited for communication but harder to control precisely. The diversity of approaches is itself a sign of a field in rapid development, exploring the space of solutions rather than converging prematurely on a single path. The question of validation is also central. When we build a quantum simulation of a neural system, how do we know it is correct? For small systems where classical computation is still possible, we can compare the quantum simulation result to the classical result. For the large systems where quantum simulation is the only option, there is no classical benchmark. Validating a quantum simulation of a neural circuit that is beyond classical computation requires new experimental strategies. One approach is to make predictions from the simulation and test them against recordings from real neural tissue. Another is to use mathematical properties of quantum systems, such as conserved quantities, to verify that the simulation is behaving correctly internally. Developing these validation methodologies is an active area of research, running parallel to the hardware and algorithm development. There is also an emerging field that connects quantum biology directly to quantum computing in a feedback loop. As researchers discover more about the specific quantum processes that operate inside neurons and other biological cells, that knowledge can be used to better design quantum simulations. And as quantum simulations generate predictions about biological quantum dynamics, those predictions can be tested in experiments on living cells. This iterative loop between theory, simulation, and experiment is the same process that has driven progress in all areas of physics. It is beginning to take shape in the emerging field of quantum neuroscience. The researchers at the forefront of this field come from extraordinarily different backgrounds. Quantum physicists who have spent careers understanding superconducting circuits or trapped ions are collaborating with neuroscientists who have spent careers recording neural activity in living animals. Chemists who understand protein folding are working with computer scientists who design quantum algorithms. The collaboration across these disciplines is not always easy. The languages are different, the time scales are different, the culture of progress is different, but the convergence is real and productive. The level of investment from both public funding agencies and private enterprise reflects the seriousness with which the scientific community is taking this convergence. The United States Department of Energy has funded quantum simulation programs specifically aimed at biological applications. The European Union's Quantum Flagship program has included quantum simulation of complex systems as a priority area. Private companies including Google, IBM, and Microsoft along with a growing ecosystem of quantum startups, have all made quantum simulation a central part of their development roadmaps. The involvement of commercial entities reflects a recognition that the applications of quantum simulation extend well beyond pure scientific understanding. Drug discovery is a trillion-dollar industry genuinely limited by the accuracy and speed of molecular simulation. Brain-computer interfaces are a rapidly growing field genuinely limited by decoding latency. Neurological diseases affect hundreds of millions of people worldwide and represent an enormous unmet medical need. The commercial incentives are aligned with the scientific goals in a way that is relatively rare in science and technology. This alignment accelerates progress. The dopaminergic system in particular is intimately connected to some of the deepest questions in neuroscience. Dopamine is often described simply as the brain's pleasure chemical. Contemporary neuroscience understands it more precisely as a reward prediction error signal. Dopamine neurons fire not when something good happens, but when something better than expected happens. If the brain predicts no reward and a reward appears, dopamine neurons fire strongly. If the brain predicts a reward and the reward arrives exactly as predicted, dopamine neurons barely fire. If the brain predicts a reward and it does not appear, dopamine neurons are suppressed below their baseline firing rate. This reward prediction error signal drives learning. It teaches the brain what patterns of action lead to better outcomes than expected, so those patterns can be reinforced. It is the engine of habit formation, skill acquisition, and motivated behavior. This system is exquisitely sensitive to the precise timing and magnitude of events. A change of a few milliseconds in the timing of a reward signal, or a change in reward quantity smaller than can be detected consciously, alters the dopamine signal and changes what the brain learns. The quantum mechanical sensitivity of the molecular machinery driving dopaminergic neurons may be part of what allows this system to detect such fine-grained differences. A quantum simulation of the dopaminergic system would allow researchers to test this hypothesis directly. Does the quantum noise in the molecular machinery create variability in the reward prediction error signal that corresponds to the observed variability in human learning? Does disrupting specific quantum mechanical steps in the signaling cascade alter the precision of reward learning in ways that match the symptoms of conditions like addiction, depression, and schizophrenia? The NMDA receptor, a specific type of glutamate receptor, plays a central role in how the brain stores memories. NMDA receptors act as coincidence detectors. They open only when two conditions are met simultaneously. The presynaptic neuron must release glutamate, and the postsynaptic neuron must already be depolarized by other inputs. This AND gate property is what allows the synapse to detect correlated activity. When the NMDA receptor opens, it allows calcium to flow into the postsynaptic cell, triggering the intracellular cascade that ultimately leads to protein synthesis and synaptic strengthening. The opening of the NMDA receptor requires not just the binding of glutamate, but also the removal of a magnesium ion that normally blocks the channel. This magnesium block is relieved only when the postsynaptic membrane is sufficiently depolarized. The precise timing of this event is determined by the quantum mechanical dynamics of ion binding and channel conformation. A quantum simulation of NMDA receptor function would generate predictions about the precision of synaptic coincidence detection that could be tested against electrophysiological measurements. It would also generate predictions about how specific mutations in the NMDA receptor, many of which are associated with schizophrenia and autism spectrum disorder, alter the coincidence detection process. The brain does not run at a fixed operating point. Its computational properties change continuously depending on the levels of neuromodulatory chemicals like dopamine, serotonin, norepinephrine, and acetylcholine. These chemicals are released by small clusters of neurons in the brainstem and diffuse across large volumes of brain tissue, modulating the responses of billions of neurons simultaneously. They set the tone of neural processing. They determine whether the brain is in a mode of focused exploitation of known solutions or open exploration of novel possibilities. They regulate the speed of learning, the threshold for action, the salience of reward, and the capacity for sustained attention. Every step of the neuromodulatory cascade involves quantum mechanical processes in proteins. The binding of a dopamine molecule to its receptor involves quantum mechanical recognition between molecular surfaces. The activation of the downstream signaling enzyme involves a conformational change driven by electron transfer. A quantum simulation of neuromodulatory signaling would be a scientific milestone in its own right. Many of the most important drugs in psychiatry work precisely by altering neuromodulatory signaling. Antidepressants modify serotonin levels. Antipsychotics target dopamine receptors. Stimulants enhance norepinephrine and dopamine signaling. We design these drugs through a combination of informed guesswork and clinical trial and error. A quantum simulation capable of modeling the full molecular cascade of neuromodulatory signaling would transform psychiatric drug development from art to engineering. The mainstream discussion of quantum computing almost always skips over a question that turns out to be fundamental. What is the brain actually optimizing for? Classical computers are designed to find correct answers to well-defined problems. This is enormously useful, but it is not obviously what the brain is doing. The brain did not evolve to solve calculus problems. It evolved to keep an organism alive in a complex, uncertain, rapidly changing environment. That goal requires a completely different computational strategy. One of the most striking features of neural processing that reflects this imperative is predictive coding, the prevailing model of visual processing used to be feed-forward. Light hits the retina, signals propagate through a hierarchy of visual areas, and somewhere at the top of the hierarchy an object is recognized. This model is wrong, or at least deeply incomplete. The brain's visual system is not primarily a bottom-up signal processor. It is a prediction machine. At every level of the visual hierarchy, higher brain areas are sending predictions downward, anticipating what the lower areas are about to signal based on prior experience and current context. The lower areas compare the incoming sensory signal with the prediction from above. If the signal matches the prediction, almost nothing happens. If the signal deviates from the prediction, a prediction error is sent upward. Only the unexpected generates strong neural activity. The brain is running a constant simulation of its environment and updating that simulation based on deviations from prediction. This predictive coding framework means that the brain is not primarily processing reality. It is constructing a model of reality, verifying it against incoming data, and updating it when wrong. The subjective experience of perceiving the world is, in this framework, largely a simulation generated by the brain's internal model, with sensory data serving mainly as error signals that correct the simulation when it goes off course. A quantum simulation of the brain must therefore model not just the passive processing of sensory signals, but the active generation of predictions. The Bayesian brain hypothesis, which generalizes predictive coding to a full statistical framework, argues that the brain is essentially implementing Bayesian inference. Given a prior distribution over possible states of the world and incoming sensory data, the brain computes the updated probability distribution over world states given the data. This is optimal inference under uncertainty. It is also computationally expensive for a classical system, because exact Bayesian inference in high-dimensional spaces is exponentially hard. The brain must be implementing some efficient approximation. One intriguing hypothesis is that the brain implements approximate Bayesian inference using quantum sampling. Quantum systems naturally sample from probability distributions defined by their wave functions. If the brain's quantum dynamics effectively implement a quantum sampling process over the space of possible world states, The result would be approximate Bayesian inference at biological speed and efficiency. This would explain both the brain's remarkable efficiency and its characteristic pattern of errors and biases in perception and decision-making. The brain would not be calculating the Bayesian posterior exactly. It would be sampling from a quantum approximation of it. This hypothesis makes specific predictions about the statistics of perceptual decisions that could, in principle, be tested against experimental data. The connection to quantum biology runs deeper than the brain alone. Every major biological system we look at closely enough reveals quantum mechanical features that contribute to its function. Photosynthesis uses quantum coherence to route energy with near-perfect efficiency from light-harvesting complexes to the reaction center. The navigation of migratory birds may exploit quantum entanglement in cryptochrome proteins to sense the direction of Earth's magnetic field. The sense of smell may involve quantum tunneling of electrons in odorant molecules. In each of these cases, evolution has not been limited to the crude material of classical mechanics. It has found mechanisms of efficiency and sensitivity at the quantum level that classical processes cannot match. The brain, as the most complex biological organ and the one subject to the most intense evolutionary selection pressure, should be expected to have exploited quantum mechanics more extensively and more sophisticatedly than any other biological system. The evidence is accumulating that it has. Every night, the human brain undergoes a process of dramatic reorganization. Synaptic connections that were active during the day are selectively pruned or strengthened. New proteins are synthesized at specific synapses. Waste products that accumulate during waking activity, including misfolded proteins like the beta amyloid associated with Alzheimer's disease, are cleared from the brain tissue through a system of fluid channels that is more active during sleep than during wakefulness. Memory traces formed during the day are consolidated through a process of replay, in which the patterns of activity that encoded a new experience are reactivated during slow-wave and rapid eye movement sleep. The molecular mechanisms by which specific memories are selected for consolidation during sleep and others are allowed to decay are unknown in detail. The relationship between the oscillatory rhythms of sleeping brain activity and the molecular processes of synaptic modification that occur during those rhythms is known only in broad strokes. A quantum simulation of neural dynamics during sleep would be one of the most scientifically valuable experiments that could be run on such a platform. Sleep disturbances are associated with virtually every psychiatric and neurological condition. Chronic sleep deprivation is one of the most reliable ways to impair cognition and increase the risk of mental illness. The accumulation of amyloid during periods of insufficient sleep is one of the most compelling known risk factors for Alzheimer's disease. Consciousness, whatever it is absent during dreamless sleep. The brain is still active during this phase, still running its molecular machinery, still clearing waste and consolidating memory, but the lights are off. Something that is present during waking and during REM dreaming is absent during dreamless sleep. If quantum simulation can identify what physical process, what pattern of quantum dynamics is present in the conscious state and absent in the dreamless sleep state, that identification would be among the most significant findings in the history of neuroscience. It is not asking the abstract philosophical question of what consciousness is. It is asking the physically precise question of what changes between the waking state and the dreamless sleep state that could account for the difference in conscious experience. The circadian rhythm that governs the sleep-wake cycle is itself regulated by molecular processes that are explicitly quantum mechanical. The core of the circadian clock is a transcription-translation feedback loop involving proteins with precise binding geometries. Mutations in circadian clock genes cause severe rhythm disorders, and these disorders are associated with increased risk of cancer, metabolic disease, psychiatric illness, and neurodegenerative disease. A quantum simulation of the molecular circadian clock would generate predictions about how specific mutations alter the timing and robustness of the oscillation. These predictions could be tested in cell cultures, carrying the specific mutations using fluorescent reporters of clock gene expression to track the oscillation in real time. The accuracy of the circadian clock is itself a remarkable physical phenomenon. A molecular oscillator involving a small number of protein molecules oscillating through a transcription-translation cycle that takes approximately 24 hours manages to keep time with a precision that the most accurate classical mechanical clocks took decades of engineering refinement to achieve. The molecular noise that would be expected to cause drift in such a small molecular system is somehow suppressed. Whether quantum coherence plays a role in this noise suppression is an open question that a quantum simulation could address. Our intuition tells us that the brain is a classical machine because the brain is large, warm, and operates on time scales we can perceive. But the physics says otherwise. The physics says that the processes underlying cognition are quantum mechanical at their foundation. And if history is any guide, the physics is right and the intuition will need to be revised. Every major scientific revolution has involved discovering that the world is stranger and more interesting than our intuition suggested. Classical mechanics suggested a deterministic clockwork universe. Quantum mechanics revealed a universe of probability and uncertainty. Special relativity revealed that space and time are not absolute but relative to the observer. General relativity revealed that gravity is not a force but the curvature of spacetime. Each of these revelations required abandoning intuitions that had felt certain and replacing them with frameworks that were initially counterintuitive but turned out to be physically accurate. The quantum brain simulation program is following the same logic. What comes after the revision is hard to predict in detail, but the general direction is clear. We will have a more accurate physical picture of what the brain is. We will have tools capable of simulating that physics with unprecedented accuracy. We will be able to ask questions about cognition, consciousness, and mental health that are currently unaskable. And we will have to grapple with new ethical questions about the moral status of systems that implement the physics of thought. We began by noticing an energy gap that the standard model of computation cannot explain. A 20-watt brain outperforming a megawatt supercomputer on the task of being itself. That gap was the first signal that the classical framework was missing something. We have traced that observation through the biology of the neuron, the thermodynamics of computation, the quantum behavior of biological molecules, the architecture of quantum processors, the breakthrough in error correction, and the applications ranging from drug discovery to brain-computer interfaces to the fundamental question of consciousness. The quantum framework does not erase the mystery, but it gives us a language capable of engaging with it. The brain speaks in probability, superposition, and entanglement. For the first time, we are building machines that speak the same language. The qubits are getting cleaner, the error rates are falling, the algorithms are becoming more sophisticated, and the biology is revealing its quantum architecture more clearly with every experiment. What will be learned from quantum simulations of the brain, even at reduced resolution, has the potential to be scientifically transformative. A real-time simulation provides a platform where you can observe every component at full resolution and full speed, run controlled experiments that would be ethically impossible in a living subject, and test theoretical models of cognition against a system that obeys the same physical laws as biology. What is still genuinely unknown is where the quantum signature in the brain most decisively lives. The evidence for quantum processes in microtubules is suggestive but not yet conclusive, demonstrating unambiguously that quantum coherence persists long enough in a warm biological environment to influence neural computation requires measurements at the boundary of what current instrumentation can achieve. This is not a reason for skepticism. It is a reason for more careful science. What is also still unknown is how consciousness relates to the physical substrate. Integrated information theory is one framework. Global workspace theory is another. Predictive processing models offer yet another angle. None of them is definitively proven. None of them is definitively wrong. The hard problem remains hard, but the tools we are building will generate data that bears directly on these questions in ways that pure philosophical analysis cannot. A quantum simulation of a brain, sufficiently detailed and sufficiently stable, will be a mirror that reflects not just the mechanics of cognition, but the question of what cognition actually is. We are not just building a better tool. We are building a tool that will force us to confront questions we have previously been able to defer. Questions about what computation is. Questions about what experience is. Questions about where the boundary between model and reality lies. The brain is the universe's most intricate attempt to understand itself. We are finally building the tools that may help it succeed. The gap between classical computing and the brain has been visible for decades. We measured it in watts, in synapses, in seconds of simulation time. We diagnosed it as a fundamental incompatibility between the physics of silicon and the physics of biology. Now we are building, step by step, the tools to bridge it. The bridge is under construction. And what waits on the other side? A genuine scientific understanding of the physical basis of mind is among the most profound destinations that the human drive to understand has ever aimed for. If you found this exploration of quantum mechanics and neuroscience worth your time, consider subscribing. The next results from quantum hardware will be significant. The next discoveries about quantum effects in biological systems will be significant. This is an area where news from the laboratory genuinely changes what is possible to think. That is a rare thing, and it is happening right now. There is a dimension of this story that connects the physics of quantum computation to something broader than neuroscience, something that touches the history of how physics itself has changed the way humanity understands its place in the universe. Each major shift in our physical understanding has not just altered our theories, it has altered what questions we thought we were allowed to ask. Before quantum mechanics arrived as a formal theory in the 1920s, the universe appeared to be a mechanism. Given sufficient knowledge of the starting conditions, the future was in principle calculable. Determinism was not a philosophical position. It was an implication of the physics. Free will, consciousness, the sense that choices are genuinely open rather than predetermined, these things had no obvious home in a deterministic clockwork universe. The founders of quantum mechanics, Niels Bohr, Werner Heisenberg, Erwin Schrödinger, Max Born, found themselves confronting something genuinely strange. The universe, at its most fundamental level, refused to be deterministic. Probabilities were not expressions of ignorance. They were the fundamental nature of physical reality. The measurement problem, the question of what actually happens when a quantum system interacts with a measuring device and a definite outcome appears, has never been fully resolved. Different interpretations of quantum mechanics give different answers. The Copenhagen interpretation says that the wave function collapses when measured, but says nothing about what constitutes a measurement or why collapse happens. The many worlds interpretation says that the wave function never collapses, and that every possible outcome actually occurs in a branching structure of parallel realities. Relational quantum mechanics says that quantum states are defined relative to observers, not absolutely. The pilot wave interpretation restores a kind of determinism, but at the cost of introducing a non-local guiding field. These are not equivalent interpretations that agree on all predictions. They disagree about the deep structure of reality, and no experiment has yet been designed that can definitively distinguish them. What connects this unresolved debate to quantum brain simulation is this: if the brain is a quantum system, then the measurement problem is not just a puzzle in physics. It is a puzzle that includes the brain itself as one of the systems doing the measuring. The brain is the device through which the physical world is observed. If consciousness is somehow implicated in the collapse of the wave function, as some early interpretation suggested, or if the brain is itself a quantum system whose states are genuinely indeterminate until some further interaction occurs, then the physics of measurement and the physics of mind are not separate topics. They are aspects of the same problem. This connection is not widely accepted in mainstream physics. Most physicists regard consciousness as irrelevant to the measurement problem and believe that decoherence, the process by which quantum systems lose their coherence through interaction with their environment, provides a satisfactory account of why macroscopic systems behave classically. But the hard problem of consciousness, the question of why any physical process gives rise to subjective experience, is not resolved by decoherence. Decoherence explains why quantum superpositions become effectively invisible at the macroscopic scale. It does not explain why there is something it is like to observe the result. This is the territory into which quantum brain simulation is headed. Not immediately, and not with certainty of arriving at answers, but the trajectory of the research points toward questions that have been considered intractable, and toward experimental tools that may finally make those questions answerable in a physically precise way rather than a purely philosophical one. Consider what it would mean to have a quantum simulation of a neural system undergoing anesthesia. The simulation would be running the same quantum dynamics as the biological system. At the moment of consciousness loss, there would be a specific change in the quantum state of the simulation, a specific pattern of quantum dynamics that disappears. If that pattern could be identified, it would constitute the strongest empirical constraint ever placed on theories of consciousness. Theories that predict the correct pattern would survive. Theories that predict a different pattern would be falsified. This is how science resolves questions, not by philosophical argument but by comparison with evidence. The same logic applies to other transitions between states of consciousness. The onset of dreaming during rapid eye movement sleep involves a dramatic change in the pattern of neural activity. The brain that is dreaming is not the same physical system as the brain in dreamless sleep. The differences are measurable in the firing patterns of neurons, in the levels of different neuromodulators, in the synchronization patterns across brain regions. But the differences at the quantum level, the specific changes in the quantum dynamics of the molecular machinery, are not yet measurable. A quantum simulation that accurately models both states would make those differences explicit and quantitative. It would say, this specific change in the quantum dynamics corresponds to the onset of dreaming. Test this prediction in a living system by looking for this specific signature. This is the promise of quantum neuroscience as an experimental science. Not speculation about consciousness, but concrete, testable predictions about the physical correlates of specific states of experience. The predictions are quantum mechanical, which means they are not predictions that classical neuroscience could make or test, but they are predictions that quantum biology experiments, operating at the frontier of measurement sensitivity, can in principle address. The development of quantum sensing technologies is relevant here. Quantum sensors exploit quantum mechanical effects to achieve measurement sensitivity far beyond what classical instruments can achieve. Nitrogen vacancy centers in diamond, for example, are quantum systems that are exquisitely sensitive to local magnetic fields. They can detect the magnetic field generated by a single electron spin. This sensitivity is many orders of magnitude beyond what conventional magnetometers can achieve. Nitrogen vacancy sensors are being developed for biological applications, including the sensing of magnetic fields generated by ion currents in neurons. If this technology matures, it may become possible to measure the quantum mechanical activity of individual ion channels in a living neuron, in real time, without disturbing the biological system. This would represent a revolutionary advance in experimental neuroscience. Currently, recording the electrical activity of a neuron requires placing an electrode in physical contact with the cell, which is invasive and limited in the number of cells that can be simultaneously recorded. a quantum sensor that can detect the magnetic signature of ion channel activity from outside the cell, and do so for many cells simultaneously, would transform our ability to observe the brain at the level of its fundamental physical processes. Combined with quantum simulations that predict what those physical processes should look like, the result would be a feedback loop of unprecedented scientific power. Predict with the simulation, measure with the quantum sensor, compare, refine. The path from where we are today to that level of capability is long and uncertain in its details, but the direction is clear. The physics supports it. The early experimental results support it. The engineering trajectory of quantum hardware, quantum sensors, and quantum algorithms supports it. What we are watching is the early stages of a transformation in how science studies the most complex physical system we know of. There is another angle on this that deserves attention, and it involves the concept of emergence. In physics, emergence refers to phenomena that arise at one level of description from interactions at a lower level, but that cannot be predicted from the properties of the lower level in isolation. Superconductivity is an emergent phenomenon. Individual electrons do not superconduct. But at low temperatures, the collective quantum behavior of many electrons gives rise to a state of matter with zero electrical resistance, a property that simply does not exist at the level of individual particles. Consciousness may be an emergent phenomenon in a similar sense. Individual neurons do not seem to be conscious. Individual synapses do not seem to be conscious. But the collective quantum behavior of hundreds of billions of neurons, interacting through trillions of synapses, may give rise to a state of physical organization that has properties, including subjectivity and unified experience, that do not exist at the level of individual components. If this is the right picture, then the quantum simulation program is aimed at understanding the collective quantum dynamics of a system large enough for consciousness to emerge. This is a physically well-posed question. We know how to think about emergence in quantum systems. We know what kinds of collective quantum states arise in other condensed matter systems. We do not yet know whether the collective quantum dynamics of the brain produce an emergent state of the relevant kind, but we now have theoretical frameworks and, increasingly, experimental tools for investigating this. The quantum simulation of the brain, understood through the lens of emergence, becomes a search for new phases of quantum matter. The brain may represent a phase of quantum matter that does not occur anywhere else in nature, a phase characterized by the specific quantum dynamics of interacting biological molecules at physiological temperatures, organized in a way that has been refined by billions of years of evolution. If such a phase exists and has emergent properties that include the capacity for subjective experience, then identifying and characterizing that phase would be a discovery of fundamental importance, not just for neuroscience and medicine, but for physics itself. This is the sense in which the quantum brain simulation program connects to the deepest questions of physics, not just the physics of neurons, but the physics of what kinds of ordered states are possible in quantum systems, and what properties those states can have. The universe has apparently produced at least one physical system capable of contemplating itself. Understanding the quantum mechanical basis of that capability is not a narrow specialized problem. It is a problem about the range of what matter can do. We are living at an unusual moment in the history of science. The tools required to approach these questions are becoming real at the same time that the theoretical frameworks for thinking about them are becoming sufficiently precise to generate testable predictions. The hardware advances of the past few years, including the demonstration of exponentially improving error correction in quantum processors, have moved the technology from theoretical possibility to engineering roadmap. The biological discoveries of the past 2 decades, including the observation of quantum coherence in biological systems, have moved the hypothesis of quantum brain function from speculation to a serious scientific program. The theoretical advances in understanding consciousness through frameworks like integrated information theory and predictive processing have moved the question of consciousness from purely philosophical territory towards something that can, in principle, be empirically constrained. These three developments, hardware, biology, and theory, are converging simultaneously. This convergence is not a coincidence. It reflects the fact that the questions being asked are real questions about nature, and that real questions, pursued carefully over long periods, eventually accumulate the knowledge needed to address them. Science does not move at a uniform pace. There are long periods of incremental progress punctuated by moments of rapid transformation, when multiple converging lines of investigation suddenly make a previously intractable problem tractable. We may be entering such a moment in the science of the brain. The consequences of success would be profound and wide-ranging. In medicine, a physically faithful simulation of the human brain would transform our ability to understand and treat every condition that involves the brain. Alzheimer's disease, Parkinson's disease, schizophrenia, depression, autism spectrum disorder, traumatic brain injury, the mechanisms of addiction, the biological basis of personality, all of these would become accessible to a level of mechanistic understanding that is currently impossible. Drug development would shift from a process of educated trial and error to something resembling precise molecular engineering. The development of effective treatments for conditions that currently have none would be dramatically accelerated. In technology, the insights from quantum brain simulation would feed back into the design of quantum computers themselves. The brain is the most sophisticated information processing system we know of. Understanding how it processes information at the quantum level would provide a design template for quantum computing architectures that is grounded in billions of years of evolutionary optimization. Neuromorphic quantum computing, computing architectures that implement the quantum dynamics of biological neural networks, could represent the next generation of computing hardware, combining the energy efficiency of biological computation with the programmability of engineered systems. In philosophy, the empirical constraints generated by quantum brain simulation would force a reckoning with questions about the nature of mind that have been debated for centuries without resolution. The question of whether consciousness is a purely physical phenomenon, and if so, what physical phenomenon it is, would move from the domain of speculation to the domain of evidence. The answers, when they come, will not be simple. They may well reveal that our current concepts of mind, matter, and causation require revision, but they will be grounded in physics rather than in argument, and that grounding will make progress possible in a way that purely philosophical analysis has not managed to achieve. The universe is made of quantum mechanics. The brain is made of the universe, and we are finally building computers made of the same physics as both. Whether that shared physics will eventually allow us to understand what the brain is doing when it thinks, feels, and experiences, and what it means that any arrangement of matter can do these things at all, remains the great open question at the frontier of this program. The energy gap we started with, the 20-watt brain outperforming the megawatt machine, was never just an engineering puzzle. It was a signal from nature that we were missing something fundamental. The quantum framework is our best current candidate for what that something is. The early evidence says we are on the right track. The road ahead is long, the questions are hard, and the answers will matter more than almost anything science is currently pursuing. The tools are becoming real. The work is underway. Pay attention to what comes next. One aspect of this scientific program that has not yet been fully appreciated in public discussion is how it changes the relationship between neuroscience and the rest of physics. For most of the history of science, these fields have been essentially separate. Physics occupied one conceptual territory, the behavior of atoms, particles, fields, and forces at scales ranging from subatomic to cosmological. Neuroscience occupied an entirely different territory, the behavior of cells, circuits, and systems that give rise to perception, cognition, and behavior. The methods were different, the language was different, the experimental apparatus was different, and the questions being asked were different. This separation was never complete. Biophysics has always existed at the boundary, applying physical tools to biological problems. Computational neuroscience has long used mathematical frameworks from physics to model neural dynamics, but the core research programs remain distinct, and researchers in each field could spend entire careers without engaging seriously with the other. Quantum brain simulation collapses this separation. It requires not just a collaboration between quantum physicists and neuroscientists, but a genuine conceptual integration. The question of how a neuron fires is not just a biological question. It is a quantum mechanical question. The question of how memories are stored is not just a cognitive question. It is a question about the quantum dynamics of molecular machines. The question of what consciousness is, the hardest question neuroscience has ever faced, cannot be answered within neuroscience alone if the answer involves quantum mechanical processes that classical neuroscience cannot detect or model. This integration is happening in research programs, in funding initiatives, and increasingly in the training of a new generation of scientists who are fluent in both quantum mechanics and neuroscience. The emergence of quantum neuroscience as a recognized field, with its own journals, conferences, and research programs, represents a structural change in how science is organized around these questions. The history of science suggests that the most significant advances often happen precisely at the boundaries between established fields, where the tools and concepts of one domain are applied to the questions of another. Molecular biology emerged from the application of X-ray crystallography and chemical methods to biological questions. Cognitive neuroscience emerged from the application of imaging technologies and cognitive psychology to questions about brain function. Quantum neuroscience is following this pattern, applying the tools and concepts of quantum mechanics to the deepest questions about the brain. The timeline for progress in this field is uncertain. Some of the near-term milestones, like demonstrating quantum advantage for specific molecular simulation problems relevant to biology, may be achieved within this decade. Others, like the real-time quantum simulation of a complete cortical column with full biological fidelity, will require hardware that does not yet exist and will likely take much longer to achieve. The simulation of a full human brain at the quantum level is a goal for a future that no one can date with confidence. But the trajectory is clear, and the intermediate milestones are scientifically valuable in their own right. A quantum simulation of a single ion channel would transform our understanding of neurological diseases caused by ion channel mutations. A quantum simulation of a single synapse would transform our understanding of the molecular basis of learning and memory. A quantum simulation of a small neural circuit would provide data about quantum correlations in neural dynamics that has never before been accessible. Each step produces knowledge and enables the next step. The progression is not speculative. It follows from the physics, the available algorithms, and the trajectory of hardware development. The scientists working in this field often describe a sense of being at a genuinely unusual moment. The combination of quantum hardware capable of maintaining coherent states long enough to run meaningful simulations, experimental techniques in quantum biology sensitive enough to detect quantum effects in living cells, and theoretical frameworks connecting quantum mechanics to neuroscience precise enough to generate testable predictions. This combination has not existed before. The questions have always been there. The tools are becoming real. What motivates the people doing this work is not just the prospect of better medicine or better technology, though both of those are genuine and compelling. What motivates them is the same thing that has always motivated science at its most ambitious: the possibility of understanding something that has never been understood before. The brain is the most complex physical system we know of. It gives rise to everything we experience: everything we think, everything we know. Understanding it at the level of its fundamental physics, at the level of the quantum mechanics that underlies its every process, would be among the deepest achievements in the history of human inquiry. The universe has spent approximately 14 billion years evolving physical systems of increasing complexity. Stars, galaxies, heavy elements, planets, chemistry, life, nervous systems, brains. The brain sits at the far end of this progression of complexity, the most intricate arrangement of matter that we know of anywhere in the observable universe. It is the thing that looks back at the universe and tries to understand it. We are now in the position of building machines, quantum machines that speak the language of physics at its most fundamental, and pointing those machines at the brain itself. What we find when we look will matter. It will change medicine. It will change technology. It will change philosophy. And it may well change how we understand what matter can do and what it means to be the kind of thing that wonders about itself. The energy gap between a 20-watt brain and a megawatt supercomputer was the first clue. Quantum mechanics is the answer the physics has been pointing toward for decades. The tools to follow that pointer are, for the first time, within reach. The work is serious, the physics is sound, the biology is consistent, and the engineering is advancing. What comes next in this story will be worth watching closely. Let us also consider what this research means for our understanding of evolution itself. Evolution is typically described in terms of natural selection acting on heritable variation, a process that operates at the level of genes, organisms, and populations. This is correct as far as it goes, but evolution does not invent its solutions from nothing. It works with the raw material provided by physics. Every mechanism that evolution produces must be consistent with the laws of physics, and the most sophisticated mechanisms evolution has produced appear to be ones that exploit those laws in unusually effective ways. Quantum mechanics provides capabilities that classical physics does not. The ability to be in superposition, to tunnel through energy barriers, to exploit entanglement for correlated behavior. These are genuine physical capabilities that evolution could potentially exploit if they confer a fitness advantage. And the evidence suggests that evolution has done exactly this. In photosynthesis, in the magnetic sense of migratory birds, possibly in olfaction, and potentially in the neural dynamics of the brain. This is a remarkable thing to consider. Evolution, which is often described as a blind process with no foresight, has apparently discovered quantum mechanics. Not by understanding it, obviously, but by stumbling onto molecular architectures that happen to exploit quantum effects, and then preserving those architectures because they work better than any classical alternative. The organisms that used quantum coherence to transfer energy in photosynthesis grew faster and left more descendants. The birds that used quantum entanglement to navigate found food and mates more reliably. Whatever organisms first evolved neurons whose quantum dynamics allowed more efficient information processing outcompeted those that did not. The brain, on this view, is a quantum computer that was built by evolution over hundreds of millions of years, through a process of trial and error at the level of molecular architecture, without any engineering team or design specification. It is the product of the longest optimization process that has ever taken place, operating on the most fundamental physical substrate available, quantum mechanics itself. When we build quantum computers, we are doing something that evolution did blindly, and we are doing it with understanding and intent. We know what quantum mechanics is. We can design systems that exploit it precisely and controllably in ways that serve specific goals. The brain we have inherited from evolution is extraordinary, but it is also limited by the constraints of biology, by the need to maintain homeostasis, by the vulnerability to disease, by the finite lifespan of biological components, by the slow pace of evolutionary change. A quantum computer, designed with understanding of what quantum mechanics can do, and programmed with knowledge derived from studying the brain, is not constrained by these biological limits. It can be made more stable, more scalable, more precisely controlled. It can implement quantum dynamics that mimic the brain's most effective computational strategies while avoiding the brain's limitations. This is the long horizon of the research program, not just to understand the brain, but to learn from it, and to build systems that carry its insights forward into a domain where biology's constraints no longer apply. This is speculative in its long-term details, but it is grounded in the same physics that grounds the near-term milestones. The same quantum mechanics that explains the energy efficiency of the brain, and that will eventually allow us to simulate the brain's quantum dynamics, also provides the foundation for computing architectures that go beyond biology. The quantum simulation of the brain is therefore not just a scientific project or a medical project. It is also, in the long run, an engineering project of the most ambitious kind. It is the project of understanding the most sophisticated information processing system that evolution has produced, and using that understanding to build systems that take the next step. What that next step looks like is genuinely unknown. But the direction, toward quantum mechanical information processing informed by our deepest understanding of the brain, is the most physically grounded direction available. None of what has been discussed here is guaranteed. Science never guarantees outcomes. What it guarantees is that careful, physically grounded inquiry generates knowledge. The quantum brain simulation program is as serious and as carefully grounded as any scientific program currently underway. The physics is solid. The biology is consistent. The engineering is advancing. The questions being asked are the right questions. And the fact that they are now approaching the domain of experiment, rather than remaining purely in the domain of philosophy, is what makes this moment in the history of science genuinely unusual. We are building machines made of quantum mechanics to study a brain made of quantum mechanics. The conversation that will result from that encounter between the physics of our instruments and the physics of our minds will be worth listening to very carefully. There is one more thread that connects everything discussed here to something concrete and near-term, something that is not decades away, but is happening in laboratories right now. It concerns the relationship between quantum simulation and drug resistance in neurological conditions. One of the most puzzling features of psychiatric and neurological medicine is that the same drug produces dramatically different outcomes in different patients. A medication that eliminates depression in one person has no effect in another and causes intolerable side effects in a third. This variability is not random. It reflects real differences in the molecular biology of different patients' brains, differences in the specific variants of receptor proteins they carry, differences in the efficiency of their metabolic enzymes, differences in the baseline levels of their neuromodulatory systems. Classical pharmacology handles this variability through clinical trial, through population-level studies that identify which drugs work on average, and through the slow accumulation of clinical experience about which patients tend to respond to which treatments. This process is expensive, slow, and often fails patients who do not fit the average response profile. Personalized medicine, the idea of tailoring treatments to individual patients based on their specific molecular biology, has been a goal in oncology for several decades. In neurology and psychiatry, it has remained largely aspirational because the molecular complexity of the brain has been too great for the available tools to handle. Quantum simulation changes the terms of this problem. If you can build a quantum simulation of a specific patient's molecular neurobiology, parameterized with that patient's actual genetic variants and actual receptor structures, you can test how a given drug interacts with that specific biology before administering it. The simulation would not just tell you whether the drug binds to the target receptor. It would tell you whether the drug, binding to that specific variant of the receptor, with that specific quantum mechanical binding geometry, produces the downstream signaling cascade that leads to the desired therapeutic effect in that patient-specific neurochemical context. This is precision medicine at the quantum mechanical level. It is the most physically complete version of the personalized medicine vision, and it requires exactly the kind of quantum simulation capacity that the current generation of quantum hardware is beginning to approach, at least for the smallest molecular systems. The path from simulating a single receptor to simulating enough of a patient's neurobiology to make reliable drug predictions is long, but each step along that path produces knowledge that can be applied clinically. Even a quantum simulation of a single receptor variant, accurate enough to predict binding affinity and downstream signaling, would be immediately useful in selecting which of the approved drugs for that condition would be most likely to work for that patient. The combination of genomic sequencing, which can identify the specific protein variants a patient carries, and